Hello everybody, this is Pranjal Ramburkar from grade 9th and today we have to go in the next class. We have to understand some basic concepts of grade 9. So today I will be starting with the most wanted chapter for the many of the students I know. So that's biology. And in biology, when we talk about science, so science is having basically the three branches in it, and in that three branches, first is bio, then we have physics and chemistry again. Fine. So today we are going to understand about the basic component of our life, the functional component of our life, and the fundamental component of our life. That is nothing but the small word and something complex that we are going to understand from this word, that is cell. Okay, so when we talk about cell is a structural and the functional unit of life. Why I am saying the word cell as a structural and the functional unit of life? I know that everyone maybe we all know about it and why it's so. So it's a really basic thing that we should understand, because this is only the thing that would help us to build our foundation. Okay, so the cell is a basic structural and functional unit of all living organism, and the cell biology. So when we talk about the cell biology, so cell biology is what? Cell biology is the study of cell in all aspects of its structure and function. So what we understand from cell biology is its structure and its function. That is very important thing for us. So what is structure then? So structure means what then? Cell gives structure to our body. It gives a proper structure and it functions in a way that the whole organism will function in that particular way. Okay, so basically there are two type of organism based on cell, that is unicellular and the second is multicellular. So unicellular means one cell, multicellular means many cell. Based on these two categories the cell is divided, that is unicellular and the multicellular organism. Okay, so when I talk about the unicellular organisms, the examples come up with, that is amoeba, paramecium, euglena, and etc. Okay, so the single celled organism is unicellular, whereas the many cell composition made up of that organism is multicellular, like the humans, animals, plants, protists, and etc. etc. Okay, see, when I talk about a unicellular organism, it is capable of an independent existence. This is my cell. Just consider it. So it's capable of its independent existence. It can exist in an independent way. It can perform the essential functions of its life by its own. Okay. So anything which is less than a complete structure of a cell does not ensure the independent living. So a cell, it is a fundamental unit, it is a structural unit, and it's a functional unit. So when we talk about the cell, the very important thing is that how the cell got discovered. So discovery of cell we are going to see. So discovery is what it is very important thing. So first is 1665. In 1665, the Robert Hooke was a scientist. He was a person who observed the dead cell under his microscope. So under his microscope, he kept the cork of a bark, this cork of a tree's bark, and when he saw under the microscope, he noticed that there are the compartment-like structure enclosed in that, and this compartment-like structures, honeycomb-like structures of its compartment, he called them as a cell. But one thing to be mentioned on that is it was a non-living cell. It was not a living cell, it was non-living cell. And he published his content in his book called as Micrographia. The book named as Micrographia was his book, and in this book only, Micrographia, he wrote about the content, he wrote about the things which he observed under the microscope. Now in 1674 again we have a new scientist called as Anton van Leeuwenhoek. He was a person who observed the living cells under the pond water. So in the pond water he observed some of the living cell, and that were the sperm cells. Okay, which he got to know under the microscope. Then in 1831 is the Robert Brown. Robert Brown saw the nucleus of the cell. Nucleus is the brain of the cell, it's the head of the cell. It manages all the functions and it coordinates the function in a proper and in a systematic way. After 4 years later, in 1835, Dujardin was a person who discovered the protoplasm, but the protoplasm term was given by J. E. Purkinje. Okay, so Purkinje was the person in 1839 who termed the protoplasm. Okay, he termed, and discovery was made by Dujardin in 1835. J. E. Purkinje was a person who said that the living matter present in the cell is called as the protoplasm. Living matter. So, what is the protoplasm? What is the nucleus? That we are going to see afterwards. But let's go towards first only discovery. Then in 1839 he said that the Purkinje said that it is the living matter, the protoplasm. Then in 1868 there was a person called as Huxley. So in 1868 the person called Huxley, he said that protoplasm is the physical basis of life, means the life cannot exist without the protoplasm. Okay, then after that in then we have a very important thing in addition to this, that is the cell theory. Cell theory is very much important for us to know before knowing the chapter. Okay, so when we talk about the cell theory in 1838, first we have 1838. In 1838, Matthias Jacob Schleiden. Okay, Matthias Jacob Schleiden was the person uh, who was a German botanist. Okay, in 1838, Matthias Jacob Schleiden, he was a German botanist. Okay, and what he did actually then, Matthias Jacob Schleiden, he examined. What he did, he examined the plants. He was a botanist. Now, obviously, if he is a botanist, he would be related to the plants. So he was a botanist. So he was a German botanist in 1838, and he observed large number of plants. And by observing, what he did, he came to know that the plants are composed of different kind of cells. The cells are of different kind of cells are there in a plant to form a tissue. At the same time in 1839 there was a person called Theodore Schwann. So Schwann in 1839 he was a British zoologist. Okay, and the British zoologist, he was a zoologist, deals with the animals. Okay, so in the animals he came to know that the animals are having the cells, and the thin layer which is present, and the thin layer which is present on the animal cell. Okay, this thin layer which is present on the cell, so that is called as the cell membrane. He came to know that, and this layer is what it is, the plasma membrane or the cell membrane we called it. So this plasma membrane or cell membrane or biological membrane was given by, so this plasma membrane, he told that there is a plasma membrane. Who told? Theodor Schwann. As well as he also told, Schwann also said that the plants are having again one outermost layer in addition to the cell membrane, that is called as the cell wall. It is also called as the cell wall. Okay, then Schwann also said again, his hypothesis, his theory is that the bodies of the animals and the plants, they are composed, they are made up of the cells and the products of the cell, means what things are made from the cell and from the cell. Okay, after this, in addition to the Schleiden and Schwann, together they formulated the cell theory. And this theory, however, did not explain as how new cells were formed. This theory was there. It told that the composition of cell, but it didn't told that how the new cells exist, how the cells were formed. Okay, so in 1855, a very well-known scientist, Virchow, Rudolf Virchow, was the person who explained for the first time that the cells arise from the pre-existing cell. So he gave the term omnicellula, e-cellula, and he modified the hypothesis, the theory which was given by Schleiden and Schwann, and the Schleiden and Schwann theory gave a final shape. So, this theory, the postulates of this theory, today what we understand is that all living organisms are composed of cells and the products of cells. Fine. The second hypothesis which he gave is that every cell comes from the pre existing cell. means a new cell comes from the pre-existing cell, the cell which was existing prior. And the third one is that the activities that we perform, that organism perform, is the outcome, is the total of the activities which cells perform. Okay, so cells activities that perform, so that is the outcome of the activities that we perform. Fine. Now let's have an overview of the cells, what we understood up till now. So a cell shows basically three parts in it. First is the envelope, the outer covering around it, then the nucleus, second is nucleus, and third one is the cytoplasm. Okay, so this all content we are going to see now. See, okay, envelope, nucleus and the cytoplasm, these are the main components of the cell. Okay, now animal cell is limited by a plasma membrane. There is a plasma membrane and animal cell is limited by it. Why is it limited by it? So that the whatever the enclosed portion is there inside this, means called as the protoplasm. Everything inside the cell membrane is called as protoplasm. Okay, so the it is limited by the plasma membrane. Animal cell is limited by the plasma membrane, and a typical plant cell is having a cell wall around the cell membrane. And then the cell wall is the outer boundary of the cell membrane. If we take example of onion peel cell, we can observe it. As well as cells are having a membrane bound nuclei. Okay, the cells are having membrane bound nuclei and they are called eukaryotic, whereas the organisms, the cells which lack this membrane bound nuclei, are prokaryotic. On basis of the nuclei, and on this basis we can categorize the cells in two types. First is eukaryotic and the second is prokaryotic. Okay, so eukaryotic and prokaryotic. So prokaryotic primitive type of nucleus it is having. Karyotic means the nucleus. So the proper nucleus which is there, that is in the eukaryotic, and the primitive type of the nucleus is in prokaryotic. Okay, so in both the eukaryotic and the prokaryotic, one thing is similar, that is the semi-fluid matrix which is present in them, that is called as the cytoplasm. It's something like a fluid structure that's transparent, we can say, as well as aqueous. Okay, it occupies the volume of the cell. The cytoplasm, it's the main arena, it's the main place where the cellular activities, the biological activities take place, and the reactions occurs in this cell as well. So beside the nucleus, the eukaryotic cell have other membrane bound organelles to the distinct structures called as the organelles, like endoplasmic reticulum is there, Golgi bodies, lysosomes, mitochondria, microbodies, etc. Okay, whereas in the prokaryotic they lack this. Now when we are talking about the size of the cell, the smallest cell that we have, that is mycoplasma is of 3 mu. Okay, so it is 3, it's when we talk about the 0.3 mu, so mycoplasma is of that size. But when we talk about the smallest cell of the mycoplasma is also there, it is one type of a cell, that is PPLO, pleuropneumonia like organism. Okay, so its size range from 0.1 mu. Okay, now bacteria is having the size from 3 to 5 mu. Okay, viruses are having 0.02 to 0.2 mu. Eukaryotic cells are from 10 to 20 mu, and the human red blood cell is 7.0, means 7 mu in diameter it is. Okay, red blood cells are having 7.0 mu in diameter. And the longest cell, that's nerve cell, can you just guess it? I know many of you have guessed it right. So it's 90 centimeter to 1 meter. Okay, so this is what the size of the cell is. Let's recapitulate it. Okay, now mycoplasma, it's having 0.3, PPLO 0.1, then bacteria 3 to 5 mu, and virus 0.02 to 0.2. After that, the red blood cells are 7.0 mu in diameter. Again, virus is done, I think. So yes, eukaryotic cell 10 to 20 mu, and the nerve cells, that was 90 centimeter to 1 meter. Okay, Now see, we will understand the next concepts, that is the unicellular and the multicellular we saw. Unicellular versus multicellular. Okay, so the organisms which are made up of single cell are unicellular. Examples I gave: amoeba, paramecium, chlamydomonas, bacteria, euglenia. Okay, the single cell is able to perform all the life processes of its own, like obtaining the food, respiration, excretion, growth, and reproduction. The organisms which are made up of number of cells, means many cells, are called as the multicellular organisms. Examples is fungi, except yeast. Okay, so in fungi we don't take yeast. Plants, animals, all cells are the multicellular organisms having basic structure and undertake similar basic functions. Now we are going to go towards the prokaryotic cell. Prokaryotic cell means primitive type of nucleus is there in this prokaryotic cell. Got it? So in the prokaryotic cell, they are represented by bacteria, blue-green algae, mycoplasma, PP yellow. They are generally small and multiply more rapidly than the eukaryotic cell. So then eukaryotic cell, which rapidly, what happens in this? Rapidly they increase its size. Rapidly they grow, multiply. Okay, so that are the prokaryotic cell. Their shape and their size, whatever their shape is there and the size is there, that will change. So I am just going to change the color of my board now from black to white. Okay, see that. So their shape, their size will change. Whose size will change? So the size of basically the size of the size of the prokaryotic cell. So there is the bacillus, the size is the bacillus. Again we have the vibrio, spiralium, coccus. Okay, so these are the different signs that we can see. Okay, but as we saw that it is having the outermost covering, that is called as the cell wall. Inside the cell wall there is cell membrane, but in addition to the bacterial cell, the cell envelope consists of a tightly bounded three layer structure it's having. So the uppermost is called as the glycocalyx. It is called as the glycocalyx. It is called as the glycocalyx. Under that there is a cell membrane and there is cell wall. Sorry, extremely sorry. So this is a cell wall and this is a cell membrane. Okay. So cell membrane is the innermost, then cell wall, and then glycocalyx. So glycocalyx, it is the outermost covering which is followed by the cell wall and the plasma membrane. So the glycocalyx and the cell membrane and the cell wall, they work together and form a very tough and one protective covering around the plasma membrane. When I talk about the glycocalyx, it can be of the two type. It is of a loose genital sheath, or it is a tough covering around. So see, this is a slime layer. Okay, and this is a tough covering. The slime layer or a is called this genital slime layer is there, and whereas thick and tough covering is called as the capsule. What it is called? It is called as the capsule. Then are formed of the polysaccharide. So this slime layer and the capsule, these are formed of what? They are formed of polysaccharide and peptides. So polysaccharide and peptides it is made up of. Polysaccharide means what? Poly means what? Many, and saccharide means sugar. So many sugars are there which are arranged properly in a sequence to form a polysaccharide, and they also contain polypeptides. Okay, they protect the cell against the antibiotic virus attack and enables the bacteria to stick. So this glycocalyx will help the bacteria to stick the surfaces which are slimy. Okay, to stick to them. Now the genitalis capsule glued together the cell of the many bacteria that live in the colony. Then the prokaryotes have a cell wall around the cell membrane, except in mycoplasma. Mycoplasma, they are not having the cell wall around them. So bacteria are classified on basis of the cell envelope and the Gram staining method. On basis of the cell envelope they have, and on basis of the Gram staining method. So Gram Weiss was a person who introduced Gram staining method. So there are two type of Gram positive and Gram negative. In gram positive, there is Staphylococcus and Clostridium, whereas in gram negative, it is E. coli and Pseudomonas. Okay, now I will ask you a question. This bacterial cell wall, it is made up of what? So this bacterial cell wall is made up of peptidoglycan. What I am saying, peptidoglycan. So peptidoglycan, bacterial cell wall is made up of peptidoglycan, and peptidoglycan is made up of polysaccharide and amino acids. So Archaebacteria differs from other bacteria in having a different cell wall structure, and this feature is responsible for survival in a extreme condition. Okay, so Archaebacteria is only having the different cell wall structure, and due to this feature only they survive in a extreme condition. Fine. Now one thing which I told that is common in this both, that is what? So that is the material which is present inside the cell, and that is called as the cytoplasm. Okay, so no, yeah. So there is no well-defined nucleus in this. The genetic material is basically necked, means we can see it through our eyes. Okay, this is the genetic material present. Fine, and yeah, what I am drawing this, I will also tell you that. Fine. Now see, the genetic material, this one is not properly enclosed, so that is necked, we can see it. So in addition, there is a genomic DNA. This is a genomic DNA, the single chromosome it is having, or circular DNA. Many of them have a they have a small circular DNA. Now this is my DNA, and outside this DNA I am having something. There is a small circular DNA outside this genomic DNA, and this is called as the plasmid. Plasmid is very essential. It's help in the phenotypic characters it gives to them, like resistance to bacteria. It helps in resistance to bacteria. No organelles are found in prokaryotic cell except the ribosomes. Except this ribosomes, nothing else is found. No more organelles are found in the prokaryotes. Prokaryotes have something unique, something different in form of the inclusions, cell inclusions. Okay, so the plasma membrane, we talked about the cell membrane. Now when we talk about the plasma membrane is semi-permeable. It means it allows only some material to pass through it and interact with the outside world. Okay, this is similar structure to the eukaryotes of the cell membrane. Now the special membrane is structure. Now I will draw for you. Okay, so what I am drawing this one. See, this is our cell wall and cell membrane, just consider. Okay, and this protruding part coming inside the cytoplasm, that are called as the mesosomes, which form the extensions of the plasma membrane into the cell, and these extensions can be in form of the vesicles, can be in form of tubules, or they can be something else. Okay, sacs they can be. So they help in what? They help in the processes such as the respiration, secretion, and increase in the surface area for the plasma membrane. For to increase the plasma membrane, it helps in that also. To the thickness will increase if these mesosomes are coming like this. So one time will come that it will be like this, and then plasma membrane thickness will increase. Okay, surface area for the plasma membrane. In some prokaryotes like the cyanobacteria, they are having something else instead of this membranous extension, and that is called as chromatophore. What it is called? Chromatophore. Now, in addition to this, now if we want to walk, if we want to go on a tour, what we like, if I wanted to go in the garden, okay, I want to go in the garden. So what I will use? I will make the best use of my foot for that. Okay, I will walk and then I will go on. Okay, I will walk and go on. So the human beings, the same way, in the human beings are motile. Motile means what? They can move from one place to another place, and plants are non-motile, means they stay on only one place, they stay on only one place, they don't move, their existence is sedentary. Fine. So when we talk about the bacteria, when we talk about the prokaryote, they are motile. They are having the protruding part from them, that is called as the flagella. So flagella is something structure like which is coming out from the body. So when we talk about flagella, let's understand this. So this is a basal body, this is a hook, and this is a filamentous structure which is coming out from the body of the bacteria. So instead of flagella, they have something else again, that is pilia, that is the fimbria. See that the pilia are the elongated tubular structures like this, like this. Okay, and they are made up of special protein, whereas this fimbria are the bristle like structure. These are the bristle brush like structures, we can see that bristles. Okay, these are the fibers which are sprouting out of the cell. In some bacteria, they are known to attach in the streams, and they attach to the stones over there. Now after this, we are coming to the very important point, and that is nothing but ribosomes. Okay, so ribosomes in the plasma membrane, they are 15 nanometer by 20 nanometer. They are made of 15 to 20 nanometer in the size they range between. They are having two subunits. This is the large subunit and this is the small subunit. It is in the prokaryote, it is having 50 years and it is 30 years. Together they combine and they form 70 years. Now we don't have to do here addition. Listen that carefully. 50 plus 30, it will be 80, but it is 70 years. Why? Because when this large subunit and this small subunit, they bind together here, due to this binding, their 10 AS is lost, and then they form 70 years. In eukaryotes, the case is of 60 years plus 40 years, and together they form as 80 years. Got it? Fine. Now we are here to the prokaryotics. So now I am going to draw the structures. See here. Several ribosomes attach to mRNA, messenger RNA, ribonucleic acid. Okay, so several RNA may attach to a single mRNA. Many ribosomes attach to the single mRNA. This is the messenger RNA. It is carrying the messenger, and this chain is called as polyribosome or polysome. The ribosomes of the polysome, they translate the mRNA into protein. So mRNA means the messenger ribonucleic acid is converted into the protein. It is converted into protein. The inclusion bodies, now very important point. These are the reserve material in prokaryote. They store the inclusion bodies. Examples of these inclusion bodies are the phosphate granule, the synoviation granules, the glycogen granules, the gas vacuoles, which are found in the blue-green and the purple and the green photosynthetic bacteria. These are present in inclusion bodies. Now this was all about the prokaryotic cell. I think so you understood whole concept of the prokaryotic cell. In the next video, we will be going to the next part, the eukaryotic cell, and we will be completing till the cell membrane or the plasma membrane. The transport also we will be completing till that point. So go on and watch my videos continuously so you will understand the concepts clearly. And this time we are having something else, something new. So this month is going, that's May, and so I'm going to, this time I'm in grade 9, so we are going to have the video lectures related to study, some motivational as well, and related to like public speaking as well. I'm going to think about it, but one thing that is for sure, that's the science. So I'm going to science and SST. So I'm going to make each topics video, so based on NCRT, CBSE, so and state board. Obviously in NCRT we are going to complete whole portion, and we will understand one by one chapters quickly, and it will make you easy to understand. So thank you everybody. Please subscribe my channel, give so many likes. Thank you. Have a nice day.